Hi, I'm Dial Holton, Microcalorimetry Product Manager at TA Instruments. I'd like to welcome you and thank you for joining us today at this webinar. During the webinar, you can customize your viewing interface by arranging windows to your liking. You can resize the video and slide windows independently from one another by clicking and dragging the bottom right-hand corner of each window. If you get disconnected at any time, please use the instructions you received to log back in. You can also access several different content windows by clicking the widgets at the bottom of the screen. These include speaker bios, additional file downloads, and the Q&A tool. If you need help, click the question mark widget. Please ask any questions you may have at any time during the presentation by submitting them through the Q&A window. We will answer as many of these as possible at the end of the webinar. Our guest speaker today is Nicola Garbett. Nicola earned her PhD in chemistry at the University of Kent at Canterbury, England. Her postgraduate work includes postdoctoral positions with Dr. Brad Chairs and Dr. David Graves in the departments of biochemistry and chemistry at the University of Mississippi Medical Center, the University of Mississippi, and at the James Graham Brown Cancer Center at the University of Louisville. She currently is an instructor of medicine and the assistant director of the biophysical core facility in the James Graham Brown Cancer Center at the University of Louisville School of Medicine. Nicola's research is focused on the application of biophysical approaches to the study of biomolecules and their interactions, with particular emphasis on the development of new technologies for medical diagnostics and drug development. She has authored or co-authored over 30 publications and is a co-inventor on a number of pending patent applications describing the application of differential scanning calorimetry as a non-invasive diagnostic tool for disease detection and patient monitoring. The title of Nicola's presentation today is Plasma Protein Calorimetry for Clinical Diagnostic Applications. Nicola?
Thank you, Dial, for the kind introduction. So I'd like to tell you a little bit today about a new, emerging and exciting application of differential scanning calorimetry, and that is its application to clinical diagnostics. So before I begin, I would like to make one presentation disclosure that the technology I will be discussing today is the subject of a number of patent applications shown here, on which I am a co-inventor. And I'm also a founder, shareholder and employee of a biotech startup company, Louisville Bioscience, which is commercializing this technology for clinical application. So just 2 slides to begin for those that are not totally familiar with differential scanning calorimetry or DSC. Shown here, you'll see a schematic of a DSC instrument. In the center, you see 2 chambers labeled S and R. These stand for the sample and reference chambers. So as it suggests, the sample chamber contains a sample of interest, typically a biomolecule such as protein or nucleic acid. And the chamber labeled R is the reference chamber, which contains the reference solution, typically the buffer that the sample is prepared in. These 2 chambers are surrounded by a number of jackets and heaters and held under constant pressure to prevent boil-off during heating and cooling cycles. So the chambers are heated at a very precisely controlled rate by the main heaters, and then the feedback heaters monitor the temperature difference between the cells such that they're held in thermal balance by applying or taking away power to the sample cell. So this signal, the power compensation signal, this directly relates to the heat capacity of the solutions, and that is the primary signal that's monitored in a DSC experiment and is directly proportional to the heat capacity of the solutions. So here I show a typical output called a thermogram, and you'll see here this is in the form of excess heat capacity as a function of temperature. This is shown for an endothermic reaction whereby heat is taken up by the sample, requiring that power be applied to the sample cell such that you get a positive profile shown here. If you integrate the area of the thermogram, this directly gives you the enthalpy of the reaction once normalized for the amount of material in the cell. And then Tm refers to the melting temperature, which is the midpoint of that profile. and that is where 50% of the molecules are in a folded state and 50% are unfolded, so the midpoint of that reaction. So here I just want to show one slide that first got us very excited about this application of DSC. And what you see on the left, in each panel there is shown in pink an average and a standard deviation for whole blood plasma that is directly analyzed by DSC. So that is the same in each panel. And then in the top panel, you will see a blue profile, which again is an average and standard deviation for rheumatoid arthritis plasma. In the middle, the blue profile refers to Lyme disease, and in the bottom, it shows samples for lupus. So you will see in each panel a very significant difference between the blue and pink profiles, and also as you compare the panels to each other, each of the blue profiles has a characteristic shape. On the right, I show serum protein electrophoresis profiles. So this is a standardly applied clinical diagnostic assay. And these exact same disease samples that were analyzed by DSC were analyzed by SPE. And you'll see some subtle differences in those profiles, but certainly not the significant differences we showed by DSC. And so this first suggested that DSC could have significant clinical utility. So here, in terms of the utility of DSC for clinical monitoring, clinically measuring plasma proteins and their disturbance as related to disease is an important clinical goal. And we feel that DSC provides a unique and complementary approach for characterizing the plasma proteome and these differences associated with disease states. So here I just show a number of diagnostic tools that are applied to characterize the plasma proteome, both in terms of clinical monitoring and also in terms of more basic research for biomarker discovery. So in terms of clinical monitoring, I've mentioned already serum protein electrophoresis. This is a very widely applied, FDA-approved assay. There are also a number of immunochemical assays for certain specific clinical markers, for example, PSA, CA125 for ovarian cancer. In terms of basic research, 2D electrophoresis as well as mass spectrometry have been commonly applied in terms of discovering new biomarkers of disease. So DSC really provides a different and complementary analytical approach for characterizing the plasma proteome based on very different underlying physical principles. So here I just want to return quickly to serum protein electrophoresis. You'll see in the top left panel, this is a profile for a healthy control sample, and you'll see that plasma is resolved into a number of bands, albumin, and then a number of globulin bands. And the table on the right, although the details are not significant, but you'll see that for there are certain characteristic changes, either a decrease or increase in these bands that are associated with a number of different disease states. And I show one example in the bottom left. This is a profile for multiple myeloma. And you can see, comparing to the control sample, there's a very characteristic change in the gamma globulin band. And this relates to an abnormal monoclonal antibody population which occurs in this gamma globulin region. And this is very characteristic for the presence of this disease. So DSC is similar in this respect. We are looking at these abundant proteins and changes related to disease states. So for biomarker discovery, 2D electrophoresis and mass spectrometry have been applied to characterize the proteome. This is a very complex system of over 3,000 different proteins. And so typically the most abundant proteins are removed to reveal the peptidome. And CELD mass spec has been commonly applied to this to essentially try and characterize the very low abundant biomarker changes. And as a result of these studies, the interactome has become an important concept. This is where proteins and peptides related to disease states have been found to be complex with abundant proteins such as HSA and IgG. And so another population, the so-called albuminome, specifically relates to understanding albumin and interacting partners. So in terms of a calorimetric approach, there are a number of advantages. It is sensitive to the extensive properties of the system, in other words, the mass of each component. So for a mixture such as plasma, the thermogram will reflect the amount of each component in the mixture. It is also extremely sensitive to interactions that affect thermal stability. And then proteins within the mixture will be resolved with respect to thermal stability, so in terms of enthalpy and TM, rather than size, shape, or charge, which is monitored by these other methods I've mentioned. So this provides really a new window into characterizing the proteome and provides different and complementary information for that. So in terms of applying DSC for clinical monitoring, the first very important question was to understand what we are monitoring. So here I've already alluded to the fact that the plasma itself is a very complicated mixture. There are over 3,000 different proteins. The pie chart on the left shows that 10 proteins represent 90% of plasma by weight. It's dominated by the red portion, which is over 50% of plasma is represented by albumin. The pie chart on the right shows that another 12 proteins comprise another 9% of plasma. So then this leaves the 1%, which contains still thousands of different proteins and peptides. And this is the population, which is characterized by typical biomarker studies, mass spec, and 2D electrophoresis that I mentioned. So DSC is a very different approach because we're monitoring the most abundant proteins and then interactions that are affecting them. So to understand the profile that I showed before, we first began by recording thermograms for the 16 most abundant proteins. They're shown in the 3 panels of the graph there. So I just want to highlight a couple of examples. In the bottom panel, the black trace represents HSA or albumin, which I've just shown represents over 50% of plasma. And you see a profile with a TM just above 60 degrees. To the right of that in red is a profile for IgG, and you see a major peak around 70 degrees and some higher temperature shoulders. And then in blue, there's a profile for fibrinogen, and you see a major profile around 50 degrees, and there are also some higher temperature transitions applying, and those represent the 3 domains of fibrinogen. And then one profile I wanted to highlight in the middle panel is that shown in mustard color. And again, there's a peak just above 60 degrees, and this is the profile for haptoglobin. So I'm going to return to some of these proteins later. Just want you to appreciate their position and the shape of the profiles. So in terms of understanding the plasma thermogram as a whole, what I show in the bottom panel here is if you take those individual protein profiles and weight them according to their abundance in plasma, you get the overall solid black profile shown there. And that is really represented by the dotted black profile, which is the albumin thermogram, which I showed earlier. And as I said, that's dominates plasma with over 50%, so that's a very dominant component of the overall thermogram. In red, you see the IgG thermogram. So this is roughly 4-fold lower than albumin, and so is represented to a lower extent there. And then just above 60 degrees, you'll see the mustard profile. That's the haptoglobin profile we saw before, and that has a very small abundance in plasma, so that has a much lower contribution. And then just above 50 degrees, you'll see one of the domains of fibrinogen in blue. So we took those profiles, as I said, weighted them according to their abundance and mathematically summed them. That gives the solid profile in the lower panel. But then we also made an experimental mixture at the plasma concentrations. That's shown in the top panel, where we took the 4 most abundant and the 16 most abundant, experimentally recorded the thermogram, and you see the profiles there. So you see very good agreement between the 2 panels, and also, if you recall earlier, the healthy control plasma profile I saw before. And so this really then led us to be able to assign some of the features of the control plasma I showed. So here you see labeled the small contribution of one domain of fibrinogen just above 50 degrees. The major peak just above 60 is dominated by HSA or albumin with a smaller contribution of haptoglobin. And then 70 degrees and higher temperature shoulders is dominated by IgG, also some contributions of IgA and IgM, and then some more minor contributions shown there, other domains of fibrinogen, transferrin, and so on. And so at this point, we really understood the nature of that control profile we saw. But in terms of applying DSC for clinical monitoring, we first wanted to establish how robust that profile would be to a number of variables. So we considered some common sources of variability in plasma proteome analysis. So here, just summarizing, certain differences in methodology could have an effect, the effect of sample storage and collection, demographic factors such as sex, age, ethnicity, and so on, other confounding factors associated with disease states, the effect of inflammation or acute phase, and so on. So to begin, we wanted to examine experimental variables. So here on the left, I just show an overall view of the assay. It's very direct. At the top shows that we just take whole plasma and dialyze this against a phosphate buffer. So this is to normalize solution conditions and provide a matched reference for the DSC analysis. We then recover samples from dialysis, dilute them to a suitable extent to analyze by DSC. And then these are directly analyzed in the DSC instrument, corrected for baseline, normalized to the total protein content so we can compare samples. And on the right, I show all the controls that we examined. So we looked at every step of the DSC analysis as well as sample handling. And we looked at a number of different variables and examined their effect on the thermogram. And what we found was that the thermogram was really very robust and not significantly affected. So we looked at a number of plasma concentrations, the effect of including or not including the dialysis step, using fresh or frozen samples, the effect of freeze-thaw cycles, time and temperature of storage, the heating rate of the experiment, replicate measurements, using plasma versus serum, and so on. And as I said, this was very robust to all of these factors. The next factors we wanted to examine was the effect of demographics on thermograms. So here I show that we took 100 healthy control samples representing different demographic categories, some males and females, different ages, as well as Caucasian, African American, and Hispanic samples. And then we determined the sensitivity of DSC thermograms to the effect of these different groups. So on the top left, I show an average thermogram and standard deviation for all 100 samples. And you see that we get a very reproducible profile with a modest standard deviation. In the bottom, I just show the distribution of demographic categories. And so what we found was there is a very slight effect of female versus male samples, which is really just some differences in amplitude of the major peaks. And this relates to some slight differences in the concentrations of the abundant proteins. There was a more significant effect of ethnicity, with the greatest effect being with Hispanic patients, and a smaller difference between African American and Caucasian groups. So then this provided very important demographic control profiles that we could then use to compare to disease categories. So we established that the method itself was very robust. We understand the nature of the profile. So the next most important question was, are DSC thermograms really sensitive to disease status? So here you'll recall this slide I showed at the beginning. This was, like I said, what got us first very excited about this method, because we saw very significant differences between these disease categories. But we wanted to examine this further in a number of other different diseases. So I'm just going to show a few examples that we looked at. So here are coronary artery disease samples. This was in collaboration with Mike Merchant and John Klein in nephrology at the University of Louisville. And so they were studying biomarker peptides associated with coronary artery disease in diabetic patients. And so they supplied to us 4 samples, and these were initially blinded. So we just got samples labeled ABCD, analysed them by DSC. And what we found were that we had two profiles that looked very much like our normal control profile, and two profiles that were very different and shifted and looked like we saw with the disease samples we looked at earlier. So then when we were told the sample status, we found that the two that looked very normal were patients that had essentially very minimal coronary artery disease, so that was in their control group. And then the two that were very shifted had very severe coronary artery disease. So this was very exciting because in a blinded fashion we could very clearly discriminate between these two groups of patients. So here I just show a number of autoimmune samples. So this was in collaboration with Marvin Fritzler at the University of Calgary, and also samples obtained from the Lupus Family Registry and Repository in Oklahoma. And so what we wanted to examine was whether DSC had some sensitivity within a disease group, in this case autoimmune disease. So we looked at 4 different autoimmune diseases, obtained average profiles for at least 25 samples, and compared them to a control profile. And so on the left we see the overall profiles of the 4 different coloured groups representing the autoimmunes, compared to the black profile, which is the control profile. And on the right, I show the standard deviations for those groups. In the bottom, we compared using ANOVA the probability of distinct means for the entire profile. And you can see that very clearly normal was able to be distinguished from all 4 autoimmune diseases to a high degree of probability. And then also comparing each autoimmune disease to each other, again could be distinguished with a high degree of probability. And so this really demonstrated that there was some discrimination within a group of diseases for a specific subtype. And here, one further example, cervical disease. This was in collaboration with Bill Helm and others at the University of Louisville. And so what we wanted to show here was whether DSC had sensitivity to pick up a so-called HSIL patient group, which is a high-grade precancerous cervical lesion. And this is what is primarily detected by the pap smear test and is a very important clinical group in terms of clinical intervention. And so we compared a study-specific control group, shown in black, to this HSIL group, shown in red, and showed quite clearly there was very, very clear discrimination between those two groups. And then we also wanted to examine what's shown in blue, which is an overall cervical cancer group, to see if there was discrimination, this group compared to the precancerous group. And what we found was, this is a very broad group of cervical cancer. Obviously, there are a number of different stages in there, but we again showed some discrimination, and also the fact that there was this progressive change in DSC profile with the extent of disease burden. So I want to just highlight some research from other groups. It's very encouraging to see that a number of other researchers have begun to apply DSC analysis to disease characterization. So here I'm going to show one study by Michnik and co-workers, and that was the application of DSC to study COPD. And so the graph I show here shows 3 groups, a control group, and then early and late COPD, so an increasing severity of disease. And you can see quite clearly that there are significant changes in the serum profile. And so they characterize this in terms of what they refer to as the TFM, or the first moment temperature. So this is essentially the centroid of the thermogram. And they found that there is a progressive shift as you go from the control group to then the early and severe COPD. So this again characterizes the progressive change and shift to higher temperatures of the profile. Here is one study from another group, Chagovets and co-workers, and they applied DSC to analyze not plasma but cerebrospinal fluid, so CSF, and this was in the diagnosis of glioblastoma brain tumors. And so there's a significant clinical need to be able to discriminate tumor type, grade, progression, and treatment effect in this disease. And this is typically done preoperatively by some form of diagnostic imaging. But what is found is that these preoperative imaging findings can often deviate from intraoperative findings. And so really there's a clinical need to identify new methods that can accurately diagnose these factors before we get to surgery. And so unfortunately there's no biochemical serum or tumor markers that can be monitored, but there is some evidence in the literature using CSF to determine protein profiles using proteomic methods. And these have identified markers that are associated with tumor aggressiveness and invasiveness and so on. And so they decided that potentially DSC could have some utility. And so what I show here in the next slide are 3 profiles for 3 different clinical groups. Again, really showing that there are some discrimination in profiles. So what I show is a control group. These are non-neoplastic control samples, so these may be patients that are having, for example, intracranial pressure monitoring as a result of having a head trauma. And these had a very reproducible profile shown on the leftmost shifted profile there, and a very tight standard deviation. The glioblastoma group is the one shown on the far right. This had a more diverse profile, and this they think is related to disease staging. This is something under further investigation. And then these were compared to a non-glioblastoma neoplastic sample group. So this, for instance, could be malignant meningitis associated with breast cancer, lung cancer, melanoma, and so on. And this showed very minor changes from the control profile. So there was this discrimination in identifying this glioblastoma group. And this, as I said, was using CSF samples and not plasma. So again, some good evidence of utility in this, analysing this local disease environment. And then I want to highlight 2 studies from Todnova and Code workers. So here, this is applying serum profiling to colorectal cancer patients as well as multiple myeloma. So here I show results for multiple myeloma, and you can see that there are very specific thermodynamic features associated that they are hypothesizing are related to monoclonal globulins, altered albumin content, and also a modified macromolecular interaction network. So unfortunately I don't have time to get into all the details of this, but the take-home message is that there are very specific signatures for those multiple myeloma subtypes. So again, a diagnostic profile that can be used. And then the other study they were involved with is, again, colorectal patients. So here you can see in the top panel the diverse profiles that they obtained, and they developed a way of classifying these based on the thermodynamic parameters of the profiles. So, for instance, enthalpy, temperature, and heat capacity maxima. And then finally, one study from a group in Hungary looked at the DSC analysis of human plasma in breast cancer patients. And so their study group was newly diagnosed, operable breast cancer patients without distant metastases. And they looked at preoperative serum samples and determined thermograms and found that they had utility for disease staging and grading. So I'm going to show here in the next slide the 2 most important findings. So on the left, compared to in black a control profile, you'll see that a progressive change in profile with increasing lymph node involvement. And on the right, again, this progressive shift of profile related to the tumor grade. So again, as I said, not just our group, but other groups have applied this in a number of different diseases and showed good clinical utility for DSC. So really, the utility has been clearly demonstrated, but the very most important question is really what causes the changes in the plasma thermogram, and also how can they be applied. So here I return to my initial slide again. You can see quite clearly that we saw this discrimination in profiles. But the first question was, why do we think these changes are occurring? So our first thought was, okay, DSC is very sensitive to the abundance and interactions in a sample. So we first wanted to see, are these changes related to significant differences in concentrations of components that would change in a disease? So we took the exact samples that we analysed by DSC, and we monitored, we analysed here using standard clinical assays, the abundant proteins and their levels in these samples. And what you'll see is, compared to control and all the diseases, there weren't not very significant differences in any of the abundant proteins, apart from the three that I marked there with asterisks. So that's haptoglobin, IgA, and IgM. So these were slightly elevated. So if I return to the profiles once again, these were elevated in lupus, which is the bottom profile shown there. And if you recall, when we looked at the profiles of the pure component proteins, remember there was a small profile for haptoglobin, just above 60 degrees, which is very small and swamped by the albumin profile in control plasma. But here you can see a much larger profile, and that would correspond with the increased concentration of haptoglobin. Interestingly, we don't see the dominant albumin profile just above 60 degrees, but we see a larger contribution above 70 degrees. We also saw that IgA and IgM were elevated, but these have a fairly small contribution to the 70-degree part of the profile. So our thought was, well, okay, the concentrations would not significantly affect the profile, but interestingly, we don't see this albumin transition. So our initial hypothesis was, well, perhaps there are some significant interactions with albumin that are causing it to be thermally stabilized and shifting out to higher temperatures. So this was supportive of a so-called interactome hypothesis, which contends that circulating biomarkers, proteins and peptides can enter the blood circulation, become complexed with circulating carrier proteins. If this was the case for a protein such as HSA, a very significant and abundant carrier protein, then that would have a very significant effect on the DSC profile. So to examine this, we tried to model this effect. So here, the graph on the right is just to remind you again that the albumin concentration was essentially unchanged in the disease sample. And so what we took was the control profile and the Lyme disease profile and attempted to model these using Gaussian components. And what we found was we could represent the profile by 6 Gaussian components for both the control and the Lyme sample. But what was very interesting was that 5 out of the 6 of these components were essentially unchanged between these samples, but one single component, which was present just above 60 degrees in the control sample, was significantly shifted out to around 80 degrees in the Lyme sample. And so that would then support quite clearly the interaction of some kind of disease component with albumin that would thermally stabilize it from the just above 60 degrees TM position in the control out to 80 degrees in the Lyme sample. So this is an area of active investigation in our group. This is just one hypothesis that we are examining. So this is an ongoing area, and what I wanted to just show in the last few slides was this certainly is important to understand the basis of the changes, but it's also we need to develop ways to be able to compare and classify samples. And so this is one approach we've developed in our lab, and this is a way to compare the entire thermogram rather than certain features that I highlighted in the other study. So this is to compare features in terms of amplitude as well as shape of an entire thermogram. And so what we developed here was if you have a reference group, which is defined by an average and standard deviation, you can compare a test curve shown in red to this in a number of ways. So the first part of the measure of similarity that we developed was to essentially measure the vector differences between these average curves. And so really the distance related to the standard deviation This can then be normalized and weighted over the entire profile, and you come up with a way to measure the probability of similarity or dissimilarity to this test group. This could then be combined with a linear correlation measurement, such as a Pearson's correlation, and then that would essentially compare the shapes of the profiles if you had two data sets that were normalized and paired. If they had shapes that were very similar, then they would be highly linearly correlated. We could combine this linear measure and this distance measure, and then you can get an overall level of similarity such that you can decide if a test curve is similar to a reference group or not. If you can make this kind of classification, you have a way to measure the diagnostic accuracy in terms of just standard diagnostic measures of sensitivity, specificity, and so on. So you would compare your test thermogram to a known positive disease group and a known negative control group, and be able to make a call, positive or negative, related to this test curve. So I show here just some preliminary results applying this method to DSC thermograms compared to a number of clinically established diagnostics that are circled here, so the pap smear test, mammogram, and CA-125 for ovarian cancer. And you can see, just for example, the CA-125 test has very high sensitivity but low specificity, so that gets a lot of the positive calls correct, but incorrectly gets a lot of false positives. And so the coloured shapes just represent some early results from the plasma thermogram assay, and you can see that they have very reasonable sensitivity and specificity that compares quite well to established clinical diagnostics. So I just want to show this, another way to compare thermograms, and this was also highlighted in one of the other studies, and this is to compare thermogram parameter metrics. So here we applied this in a study of type 1 diabetic patients, and this was really to look for new methods to identify type 1 diabetics in terms of their risk of renal dysfunction. Typically right now, as a diagnostic tool, you look for low levels of albumin leaking out from the kidneys into the urine. But this is a metric that is being questioned in terms of its predictive value. So we wanted to look at the utility of applying DSC. So what we looked at here was a prospective cohort of type 1 diabetic patients, and their renal function was monitored over the period of approximately 10 years. So we applied DSC to determine their profiles at the beginning of this study, when all patients had normal renal function. And then, as I said, these were patients followed for approximately 10 years, and so you would then know their ultimate clinical outcome in terms of renal function. And what we found here by the two graphs, the one on the left are patients that maintained normal renal function over this period, and the graph on the right are patients that had a decline in renal function. And so you see here on the left profiles that are quite similar and look like more like our control profile, whereas those on the right have a more higher degree of variability and have a profile that is altered from the control profile. So here in this slide, I show on the left the average curves for those two groups, and so you can see those changes in features, a decrease of the main transition and slightly higher amount of the profile at higher temperatures. And then on the right, I show a number of thermogram metrics. So in terms of area, Tmax, the maximum of the temperature profile, also TFM, this first moment temperature, width and height, as well as the heat capacity of the major albumin transition, as well as the higher temperature so-called IG transition, and then also their ratios. And so you can see a range of values for these two groups, but you can see the trends that discriminate these two groups. And what we tried to measure was what's the statistical significance of applying each of these metrics and which discriminate and which do not. And what what we found was that the amplitude of the profiles are statistically significant discriminators. And so you can see qualitatively that the DSC profiles are sensitive to these prognostic factors in plasma and can discriminate these groups. And also that the thermogram feature parameters have some utility as a diagnostic metric. So again, this is something that we and other groups are actively developing, but I think quite clearly there are ways that you can use in order to come up with metrics or approaches that can be applied to clinically discriminate groups. And so with that, I just wanted to conclude and say that a number of studies, both by our group and other groups, have shown that DSC could indeed provide a complementary approach to characterize biofluid proteomes, where the thermograms, at least in plasma, reflect the concentrations of the most abundant proteins, that DSC is sensitive to unique disease environments, and one hypothesis is that the shift in thermograms can result from interactions or modifications of these abundant proteins, which would support the so-called interactome hypothesis. As I said, this is an area of active investigation and certainly would require a lot more research. But I think all of these studies have shown that there's an increasing body of work that certainly demonstrate that DSC has very clear utility for clinical monitoring. So with that, I just wanted to acknowledge the people involved in the studies I've shown here, a number of people at the University of Louisville, Brad Chairs, Lynn, Jim Miller. I've shown studies from Mike Merchant and John Klein. Also a number of external collaborators, the Lupus Family Registry and Repository kindly supplied a number of samples for the autoimmune studies. Marvin Fritzler is an autoimmune collaborator. Dan Fish and others from Louisville Bioscience were very importantly involved in establishing these methods to be able to distinguish thermograms. And then current research support from the James Graham Brown Cancer Center and a competitive enhancement grant from the University of Louisville, and the Elsa U. Pardee Foundation, an NCI grant and a SBIR grant to Louisville Bioscience was very crucial in supporting the early development of this method. So with that, I would thank you for your attention and be very happy to take any questions you may have.
Thank you, Nicola, for that great presentation.